coming or not?
Cakap coming lah. Dah bagi tahu.
No, coming for real. I'm about to start now. They need to learn stuff.
I'm coming.
There is no take two in this, okay? I've been very clear since 6 months ago. Like you see, or you might have heard, I'm very particular about time. Very, very particular, okay? My class, all default A. If not getting A, it's your fault. It's as easy as that. You follow all the assignments, activities, and so on. So far, those who do not get A, you are the one who's problematic, okay? Not me, right? I have designed the course in such a way, the implication of final, very, very minimum to your overall marking. Because final, I cannot help you. That is highly centralized. Okay, you got the prefect and everything. But the rest of it, I can control. And that's where I want to help you. Well, to help you with 1 marks is one thing, but to enable you to learn as much as you can. Are they coming? So dia nak kami ke apa? Macam mana ni?
Yes, ke? I think start je lah.
Yang tak reti benda patut ada, tak nak ada. Dah tahu kelas pukul 8, bangun pukul 4. What time do you think I wake up? Okay. Dr. Shita, is she coming? Kamu macam mana? Is she coming? She needs to know as well. Dah tukang ajar pula yang nak kena risau. Who's undergrad? All you undergraduate? Okay. So can you make it for the afternoon session? Because we're not going to have anything on Thursday. Okay. For some reason, that dean office, I told them many times to have the schedule, everything for today. Biasalah rakyat Malaysia. Dia selective hearing. They only listen to the things that they want to listen. Right. Okay, just before we go further, maybe I should turn this slightly off. Okay, is that clear? Good. Right. Let's make it full screen. Yeah. So for the rest of the semester, before that, welcome to first class, welcome to first everything. Welcome to your first horror of the semester. Yeah. People in my class, my motivation is actually to drive you away. Because it's easy A. I don't want to give people easy A. But if you are the type like a hardened auditory organ, you can withstand that. That's good for you. Right. Okay. So for this course, you're going to learn about, I only offer this subject once a year. There is a reason for that. Because the content of it kind of modified from year to year. Not modified in a way it's not correlated. It's more in the sense that you learn the things that you have seen before, but at a greater depth. So that not only you understand the theory, the concept of the lesson, but you can actually associate with potentially the project that you are doing. Because you are final year, you're about to do your project. your project will very likely involve some kind of treatments and so on, and also to do general discussion. So the idea of learning physiology is when you have done a certain set of experiment, you got your observation, you got your data. How do you interpret this data? How do you discuss? How do you make connection with the existing literature? Or maybe somebody has not done before you, like at all. So how do you come up with your own set of discussion? Can it be a groundbreaking discovery? Or is it potentially some kind of error that you thought, oh, this happened, actually it's a false positive, honey. You need to calm down. Or is that the case? So in physiology, we're going to learn about that, right? So for this semester, in essence, you're going to learn these 5 things. So the book that I'll be using, actually I brought the book. It's very, very new, this book. It just got published in July, I think. Yeah. So these books got many editions. This is the 7th edition. Long time ago. Okay. UPM library do not have this because this just got released. Even I do not have the PDF version of this. However, dia dah ambil gambar benda ni, turn it into PDF. So nanti dia bagi. Right. Okay. We're going to focus about the biochemistry and metabolism of 2 things: the photosynthesis and respiration, in essence. Okay. So I know that people have learned photosynthesis before, but you'll be surprised. Whatever you learned is that's not even on the surface. Okay, that's not even on the surface. That you'll be like, can I go back to standard 1? Yeah, life is close somehow. Okay, for the first can I call it chapter? Yeah, for the first chapter, based on the books, it is chapter 9, 10, 11, 12, and 13. Okay, but for our chapter, this is chapter 1. Okay, so these are the general overview, the things that you're going to go throughout the semester. Right, for today, I hope I can finish up to 9.3. Okay. Before we go further, let's do a quick pretest just to judge how much you know. I know there is no ABC, but let's assume there's some ABC there. For number 1, let's answer this. What is the most photosynthetically active tissue in vascular plant? Who says A? B? C? D? No. So it's mesophil olive. So that's good. What's the thylakoid? A, B, or C? Who says A? Protein involved in photosynthesis? Nobody. Who says B? Specialized cell of leaf? Nobody. Who says C? Specialized internal membrane of the chloroplast? 1, 2. 2. Kau nak apa? Nak cucuk mata? 2, 3. How about D? Aqueous region where carbon reactions occur. So the answer is C. Right. How about number 3? What are the product of thylakoid reactions? Ooh, what's that? What's the product? Is A the product, ATP? Yeah. Who says no? You need to raise your hand. If you say no, just this is pre-test, okay? No marks given yet. Yeah, the judgment day has not come, so repent is possible. ATP, is it the product? Okay. NAD, is that a product? No. NADPH? Yes. How about sugar? No. So the answer is ATP and NADPH. NAD, it is a product, but not in chloroplast, in mitochondria. Okay. 4, the functional unit of transfer right energy into chemical energy are called, what is that? Is it photoprotein? No. Is it thylakoid? Is it H2O? Is it photosystem? Yes. Okay, photosystem. And finally, what is the principal electron donor in photosynthesis? Is it water? How about NADPH and ATP? No. NADPH is electron donor or the reductant product. Sorry, not the electron donor. NADPH is the reducing molecule that can give the hydrogen to something else. Okay, and ATP is the energy currency for the cell. Right. Okay. Why do I feel hot? That's not on. Why is it blinking? Why you PM like this? By the way, you need to be careful with some of this lecture room computer, okay? At one point, somebody jacked in the USB, the entire USB got obliterated. So you don't want that to happen to you. Okay. You're going to see a lot of terminologies and so on. So in order to properly understand photosynthesis, number one, you need to open your horizon. Open your horizon so big. Keep telling yourself, my brain is elegant. There is nothing in the universe I cannot absorb up to this point. Even though that's not the case, let's pretend that you can. Okay, because if you are being too close-minded, just thinking about, oh, I got agriculture, I got just the biology of it, just the agronomy of it, you are not going to be able to understand whatever that you're about to learn. Okay, so open your mind to be accepting to the new set of knowledge that maybe before this it's difficult to your view, but it's actually after you have made the connection, it's not that difficult actually. Okay, right. So we're going to go through this bit by bit and see what's going on. Right. So number one, you need to understand the properties of light. The thing about light is it's not like a one object and you're done with it. No. It is actually get created not from the beginning of time, but it get created through a series of interaction of particles with many things in the universe. Okay, so you can see that in the equation 9.1, it says that C equals to what symbol is that? Lambda and nu. So when you learn science, open your mind to a new set of language, Greek. So I suggest you're still young, go back, print out the Greek letter alphabet. Please memorize them all like ABC. How many letters in ABC? 26, right? Greek alphabet got some similar amount as well. If you are familiar with these letters, you're not going to get alienated when you learn science in depth later, because they're going to use this word all the time. So this is lambda, all lowercase, okay? Lowercase, and this is nu. So lambda referring to the wavelength, nu actually referring to the frequency. Okay, if you remember from your physics lesson, when you see something like this, the appearance of wave, yeah, what is meant by lambda is actually what? It's the distance between complete cycle of a wave. So starting from here ends about the same location as well. So that is your what? Is it lambda or nu? Lambda is actually the entirety of everything. So this actually is the frequency. So this is one complete cycle. So what about lambda? What about it? What does it say there? Yeah. So it is the distance between successive wave crests. So that is also the distance. So this you got one frequency, and if you go on again here, you got another one frequency here. So crest is this thing. Crest. Or in Malaybo, I think they call it, you know, crest? Some birds got crest. What's that, Emily? Jambul. You know chicken? Chicken got that little thing on the head, right? So that's crest as well. Okay. Then c is the speed of light. In the case here, the speed of light is actually unobstructed light. Light that is traveled in completely unobstructed manner. So that is the speed. So in order to get speed, you need to times the lambda by nu, the wavelength by frequency. Okay. So of course, when you talk about light, it has two properties. It can be particles, it can be wavelength. Just now you understand it in the form of wavelength, right? What about in the terms of particles? In the terms of particles, we call it photon, not proton. Photon, P-H-O-T-O-N. Photon is the fancy word for light particle. Okay, so when we talk about photon, we are actually referring to the energy of it, and the formula for light energy is the Planck constant, which is h, times by again the nu, right? No, we don't want to restart. Yep. Remember this: the longer the wavelength, the lower the energy. The shorter the wavelength, the higher the energy. So what is meant by that? When you have a set of wavelength, 1, 2, 3, 4, 5. These finish in 5 seconds, and you have another set of wavelength, 1, 2, 3, 4, maybe 5 here. So this wavelength takes 5 seconds to complete 5 cycles. So we call this as a short wavelength. The second one, wavelength as well, but it takes much longer, maybe 3 times more. 5 cycles in 15 seconds. So we call this is low energy. We call the top one as high energy. The more number of cycles that the wave can complete for the same amount of time, the higher the energy that it contains. Okay, and guess what? In nature, these waves can actually be detected by our eyes. In the case of the rainbow color, I'll give you that because we are familiar with that. If we have these two patterns, we can say that, oh, this pattern is actually somewhat look like a blue light, high energy. And then you go down the spectrum, at the end of the spectrum, you're going to see this is actually the red light. Okay, so it goes down in energy, and therefore it changes the appearance of the light as well, right? And this is something you can see by your own eyes. So next time when you look at the sky, you see that, oh, why is it so blue? Yes, that's why it is not safe for the astronaut not to be wearing astronaut suit. You see all the blue? Super high energy. If they don't wear special astronaut fancy looking suit, they're going to get fried out in the ISS station, right? But just before night, during the dusk time, when you look far at the horizon, then you see, oh, that's pretty. It looks so red. You know, it looks like you want to go along with the sun due to sadness. That kind of horizon feeling is looks super red, right? Far, far. That's why, because red is the dominant color now. Far wavelength coming all the way to your eyes. That's why. Okay, right. So the figure mentioned here, by the way, I need to use my rotan. I got two rotan here. I got this rotan, I got this rotan. So depending on my mood. So figure 9.1 here. Let's look at what is that. So this is to show the concept of light in terms of the very existence of light. If you want to go all the way in at the subatomic particle level. So light is actually the full science name of it, electromagnetic radiation. That is the most encompassing name of light. We call it light, right? Just call simply light. But scientists like to make things harder for a very fun reason to them. Why scientists call this electromagnetic radiation? The reason is light actually have two functions of wave. So it has the magnetic component of it, the one that you see going up here, and also the electric component of it. It comes together. So like actually this thing can move, but I'm not opening the PowerPoint now. It's not moving. So at any given moment, both electrical and magnetic field within a light that is propagating, they're going to be opposite from each other. When one guy is up, the other guy is flat. And then it changed the position like this. So it keep on disturbing each other. So one might wonder, how come light got these properties? Where it all started? I mean, like, how light is created? Oh, do we want to go down that path? How light is created? You want to know? You want to know? If you say yes, masuk exam. How light is created? That light here. How is it created? Remember, I keep telling you, you need to open your horizon. Energy conversion. Half mark. Oh no, quarter mark. It's permanent. Cannot rub this. Okay, that's not pretty. That's not pretty. This thing is permanent, but it says whiteboard. Okay. How light is created? Let's put it this way. You know electricity, right? Electricity. I'll give you a very, very fundamental way of looking at it, even though it's more complicated. I just want you to appreciate the concept of magnetic and electrical thing going together to create light. So you have, let's see, your AC regulator, the box here. And then you have it connected to a metal bar, right? So let's say that you set this to maybe 10 terahertz. In this metal bar, this is so not fun using this to write. In this metal bar, there is an electron, right? Everything got electron in it. You got electron, your friend, your enemy, everybody got electron in it. So this electron, if you didn't turn on the AC meter, it's not going to do anything. It's just everything static. However, the moment you turn it on, this thing will cause the electron to go back and forth within this metal bar. Okay, so this thing going up, down, up, down, up, down. Okay, the action of electron going up and down, up and down, up and down, it is causing something to happen surrounding it. What is it? Magnetic field. You have electricity going back and forth within the metal bar. Immediately surrounding it, you will start to have magnetic field. Okay, and this magnetic field, depending on the energy that comes from the source, will get smaller or bigger as time is progressing. Meaning that if this thing has a higher input of hertz coming toward this way, magnetic field is going to get bigger and bigger and bigger and bigger. And guess what? This is something that you see. So if you think about in the case of your light here, meaning that the moment you touch on your switch is heating up the metal thing inside the bulb. Electrode, diodes, LED. You see it got the little legs in it, right? So that gets heated up. That creates magnetic field around it. And this magnetic field will start to get bigger and bigger to the point you can start seeing it glows. So that's in essence how light is created. So meaning that electrical component and magnetic component of light is very interrelated. Okay, you cannot run away from it. Yeah. So maybe you have seen this before. So that's why we have something like this called electromagnetic spectrum. You see, wavelength, which is measured in the unit of nanometer, okay, has a corresponding frequency. Okay, pay attention here. This is called the full spectrum from here to here. Actually, it's even more, but we are focusing on that now. This is usually the spectrum of sunshine. Okay, you know you got your sun, which is far, far away from Earth, like what, 200 million kilometers away, or maybe more. So that sun is emitting this gigantic amount of light throughout all the universe. And that light is what we call as the full spectrum. Some of it come to the Earth, but not all manage to reach the surface of the Earth. The moment it hits the Earth, two things will prevent this solar spectrum to completely arrive at the Earth. What are those two things? Two things that prevent full spectrum of sunlight to reach Earth. Ozone and magnetic field. Remember, our Earth is protected by magnetic field. Invisible, you can't see it, but it's there. So, in that wide spectrum of sun or solar spectrum, only a small portion of it that is actually the visible light, or you know as the rainbow color, the one that we can see. Okay, and this spectrum is within this region here, petahertz, 10 to the power of 15 hertz. Remember, this is the nu, this is the lambda, and that that petahertz is the region here is corresponding to this wavelength value, meaning that within let's see here, 103 3 nanometer all the way to 3000 nanometer. Pay attention, okay, because this thing is going that way, this thing is going that way. This is the lower side of the bar, higher side of the bar, but this thing is opposite. The wavelength that you can only see is only from 400 to around 700. So just to give you some kind of appreciation here, meaning that the light itself is having background values or background properties. When you see, for example, you see the red color of light or the blue light color of light, remember that blue light color, that red light color has the innate value of lambda. In the case of light, blue light, around 430 nanometer. And it also has a corresponding hertz to it. Okay? Kind of like if you think about it, your age now. Let's say, how old are you? 25? Yeah, 25. Okay, let's say you're 25. But there is a hidden value as well, because if you're talking about 25, that means you are following the Gregorian calendar. You know, that calendar kuda. If you're talking about the lunar calendar, the Hijri calendar, you are actually not 25 years old. You are actually a lot older. Yes. So 25 times 10, 250, you are actually 26 something years old. Yeah. So you yourself, the same individual, based on Gregorian calendar, 25 years old. Based on Hijri calendar, you are 26 years old. Based on, I do not know, Buddha's calendar or something, you are 3000 days. Remember, you are the same person, but you have all these values associated with you. So it's the same as the light in here. Behind it, you have all these values depending on the perspective that you want to talk about. If you want to talk about the wavelength because you are particular about the color, you go with the wavelength here. If you want to talk about the energy amount of it, because that's the requirement of the formula here, you use the hertz. If you want to talk about the just the color, then we go with the red. Okay, so put things into perspective. Yeah. Oh, this thing. I was thinking whether to put this or not. Remember I told you about the light have dual nature, the duality of light properties, wave particle. So that actually came from an experiment long time ago by Young's. They call it Young's experiment. So the mystery here is, let's see. Okay, look at this. So there is a source of light, laser, and this laser is allowed to go through two slits. So by your logic, you should only see two bands of light on the wall at the end, right? Because originally you got only two slits. But actually, in the real experiment, that is not the case. Okay? You don't get this all the time. If you are looking at it, you get this pattern. But when you are not looking, it will start to form this light scatter effect, meaning that you get more than two bands on the wall. How come? Yeah, that's actually the part the scientists still cannot figure out. Why? So in this experiment, it tells the properties of light that can change depending on whether the reality is observing it or not. So this whole thing is called the observer effect of light. if the observer can be you, or can be detector, or can be camera, can be anything. If the observer is observing the light will behave like particle. Then that's why you get this pattern here. 2 slit, 2 bands. But when you're not looking, suddenly the light will start to scatter, meaning that the light has assumed its wavelength nature. Nobody knows exactly why. But this is why scientists has made a lot of speculation. Actually, whether or not you are engaging with something, looking at something, can actually change the fate of something. Yeah, yeah. Did you know that there are actually many examples of things not according to our common sense. For example, the time. The time that we have here on the surface is actually not the same similar time with the satellite orbiting our Earth. Okay, because the satellite is orbiting Earth maybe like, I don't know, maybe 9 times a day, meaning that the satellite is very fast. Correct. So the faster you get, the time kind of shrink, meaning that there is a discrepancy of time on Earth, time on the satellite. So that is to say, the satellite is experiencing the slowing down of time. We have advanced in 4 seconds, but the satellite has only advanced by 1 second. So this thing accumulating over the day, over the week, over the month, if it is not corrected, all cars on the planet is going to accident with each other. So the satellite is orbiting our Earth, put this correction to take into effect the dilation of space or the shrinking of time. Yeah, so that's kind of interesting, don't you think? Right? Yeah, time change actually, but it's, I can see the satellite is orbiting. Yeah, you can see it, but the time it is experiencing is different. Why? Because it's moving super fast. When it's moving super fast, it pressing on the time. The time becomes slow. Not slow for you, slow for that satellite. Meaning that when the satellite or whatever astronaut coming back to Earth, they're not aging. So if there is ever a riddle or teka-teki, anti-aging, join astronaut program. Right. Okay. Yeah. Right. Coming to the another concept about the absorption of the solar spectrum. Okay. So when you talk about photosynthesis, you are actually referring to the ability of photosynthesis to first capture the light energy to be turned into chemical energy. And this, for this to happen, a number of equipments need to be present first. So the very thing that capture light energy is chlorophyll. Okay, so chlorophyll is a pigment. It's just a pigment. You got a pigment as well. You got a pigment in your skin, right? What's the pigment in your skin? Melanin. Okay. This pigment actually have a specific amount of energy from the light that it absorbs. It doesn't absorb everything. Okay. So if you look at this figure here, the black curve that you see, this is the solar output from the sun, basically. Okay. So the sun is giving towards the earth. The most is 400, around 430. And guess what? This is the blue light. See? The blue light. And since blue is the most coming towards the earth, that's another reason why you see blue. Right? And then it goes all the way down. Okay? This thing, when you look at the level of what is received on the earth surface, It's pretty much the same. It's pretty much the same. But when you talk about the pigments, the one that is absorbing the energy here, the pigments do not share similar spectrum. This pigment is actually selectively only absorb two colors within the blue region and also within the red region. So that's why you see in many of the plant factory these days, they are utilizing the red-blue LED because scientists have known these days that blue and red are the lights that are most absorbed by the chlorophyll. Yeah. So the chlorophyll itself, if it absorbs, but nothing happened to it, it's not going to do any good. Okay. So this is another concept that you need to understand here. This is about the properties of the chlorophyll itself. The moment it absorbs light, if there is no interaction between the light coming to the pigments and the pigment is not doing anything about it, no photosynthesis can happen. Okay, so the chlorophyll, it's a molecule. So in the molecules, you know, you got all the bonds, all the electron and so on. There is a potential of energy transition. Okay, meaning that the moment the chlorophyll can absorb the energy and then something happened to the chlorophyll, that what makes photosynthesis possible due to this energy transition. So the figure here tells you just that chlorophyll, when there is no light, chlorophyll in darkness, it is what we call at the ground state. Nothing happened to it. Okay. By the way, chlorophyll is just like our blood. Okay, our hemoglobin. The difference is chlorophyll is green because the presence of magnesium in the middle of the atom. But for the hemoglobin, we got the iron in the middle. Right. The moment chlorophyll absorb the light, two things can happen depending on the kind of light. This is why to understand about the light. If the chlorophyll absorb the blue light, it will be immediately get excited. It gets excited. The chlorophyll, it gets excited. Okay. And this new state of chlorophyll, we call it higher excited state. However, if the chlorophyll absorb the red light, it too will get excited. But this state now is called lowest excited state. Okay. For the chlorophyll to do any work, it needs to get excited first. Whether it gets excited, highly excited, or moderately excited, or minimally excited, doesn't matter. It needs to get excited first. Okay? So it gets excited and something will happen to the electron of the chlorophyll. If you learn maybe in your chemistry or physics before, things that get excited will not stay excited forever. Okay, it will return back to the ground state. Correct? Why is that? Why chlorophyll needs to return to the ground state? Why can't it stay excited forever? Anybody know why? If you understand it, probably you can answer the question. Why? Is it good being excited? Put it this way: you have a neighbor, you have a friend, right? You tell a joke to your friend, or maybe your friend is so stoned, you keep tickling your friend, and your friend will start laughing. Will start laughing, ha 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 ha. What will happen to the laughter of your friend? will it continue? Will it get more? Will it subside? So your friend laughing is actually the excited state of your friend. It get laughter. There is a burst of laughter. Imagine your friend keep on until next Deepavali. What happened to your friend? It's going to break down. It's going to break down. Not to mention all the crazy hospital and everything. Right. So being excited, the burst of it, a split second of it is good because in the case of your friend, that releases the stress because she just got dumped. You want to cheer her up, right? You make her laugh. That's good to release the stress, but not too much until Deepavali because that will break your friend down. So in the case of chlorophyll, excitation is important as well, but not too long. Because if it's too long, this energy, this laughing, will break the entire membrane. You don't want that to happen. So that's why anything that gets excited, molecule, atom, or anything needs to return back to the ground state. That's why, because you don't want to incur damage to the surrounding molecules. Okay? Your friend, you think is not going to cause damage to the surrounding molecule. Try allow her to laugh until Deepavali. First, you cannot sleep because she keeps laughing. You cannot understand. Do you want to eat? Do you want to sleep? What's wrong with you? Cannot say anything else other than ha 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 ha. Okay, so then is the concept of absorption spectrum. I'll try to finish this within 20 minutes. This spectrum is absorption spectrum is for a given liquid, for a given solid or anything. There is a pattern of absorption here, like what you saw with the chlorophyll just now. Chlorophyll absorb two color, blue and red. Okay, so there is a technique used to study this. It's called spectrophotometry. Okay, so in the spectrophotometry, it's a machine. Actually, we got the machine in the physiology lab, but I think I'm highly confident it's broken because it's UPM, right? Oh, dah lah benda ni nak record kan nak cakap buka-buka kat UPM. Biarlah. It's the truth. You have your light. Your light will go through a prism. The light is equivalent to the sun, meaning that full spectrum, all the cosmic ray and so on. The prism will control the laser to be of specific wavelength. From 200 nanometer, for example, until 800 nanometer. So the prism can change, can turn. And then specific beam of specific wavelength will come out and hit the sample. And then the detector will detect because it knows how much the original source gives, and then how much has gone through the sample and what is the balance. So this will give an absorption function to the function of wavelength, meaning that, for example, 450 nanometer has been given, and apparently 80% of that has been absorbed, meaning that 80% of 450 nanometer has been absorbed by this particular substance, in our case, chlorophyll. Okay, yeah. Okay, you're going to see this in the books from time to time. So chlorophyll in the ground state, usually you put it CHL, just chlorophyll. But chlorophyll that gets excited due to the laser, due to the sunlight, due to the LED from the plant factory, will become excited chlorophyll, chlorophyll star. So to get chlorophyll star, you simply plus Hb. Have you seen this before? H nu. What formula is that? If you remember from the beginning, that is actually the formula for energy. Yeah. So add those two together, you will get the value of excited chlorophyll, right? Okay, so pay attention to this final sentence here. The initial photosynthetic reactions must be extremely rapid to outcompete the decay of the lowest excited state. Meaning that the moment the chlorophyll get excited, there are a set of decisions that needs to be made. Okay, pretty much that, yeah, like your laughing friend. The moment she is laughing, the body now needs to decide: do I need more? Do I need to stop now? Do I need to feel released? The brain need to decide: do I need to release more dopamine due to the laughter? Okay, following an excitation event, a number of things needs to happen. Okay, and the same thing happens to the chlorophyll. And I think most of you have learned with me before. All this while, you learn that excited chlorophyll, how many destiny can you see? 3. Now you learn 5. So I've been telling you, right? Actually, there are more destiny. I don't want to go to be crazy about it. So all this while, you learn only 3 destiny of excited chlorophyll. Excited chlorophyll meaning the electron. Where does it go? Does it go to do the photochemistry? Does it go to the fluorescence? Or is it involving the heat release? So that's actually 5. I don't know why it looks like this. Actually, it should look a lot prettier than that. Chlorophyll get excited. So what get excited actually in the chlorophyll? It's the electron. Electron that gets ejected out. Okay. The following slide, you can see there's a molecule of chlorophyll. Chlorophyll has this porphyrin ring attached to a hydrocarbon tail. This hydrocarbon tail is anchoring the pigment into the membrane of the thylakoid. Okay, that's the anchor. So when chlorophyll absorbs light, it is the functional group in a specific region of porphyrin ring that will eject the electron out. Okay, so when this electron is ejected due to the fact that the chlorophyll has absorbed some amount of light energy, it will be empty. Okay, there is a hole in it. Okay, now we are talking about the ejected electron. What happens to it? Number one, it can decides to go to fluorescence. Fluorescence simply means glow in the dark, meaning that the ejected electron, it will come back to the ground state, and then in the process, it releases photon. It releases light energy. And this light energy is usually far red. Okay, and that is fluorescence. So it gets excited, and then it just decides to return back to the ground state immediately, and from up to down to the side, glowing happening. That is fluorescence. Okay, and usually the machine measures something like 740 nanometer. 740 nanometer, our eyes can't really see. Okay, because we up to 700 only. So this is red, your infrared and so on. Yeah, that kind of red. Or the excited chlorophyll can return to the ground state without the emission of photon. Again, this guy here gets excited, go up, go down, but there is no glowing to the side. It's not being dramatic about it. There is no, sparkling of light coming, but you get the heat coming out of it. Go up, go down, heat, and this heat, you can't really see it because it's just absorbed by the surrounding molecule. Okay, Remember, this whole thing is happening in the chloroplast. Chloroplast is completely water. Okay, so the water is absorbing the heat, and usually based on calculation, this is negligible. Okay, because it's not easy to increase 1 Celsius of water. Okay. Or number 3, the chlorophyll can participate in energy transfer, meaning that the chlorophyll here get excited. This excited energy is transferred to surrounding molecule. Chlorophyll in the thylakoid membrane, it's not present alone. It's never alone. It's actually in a form of complex. We call it LHC, light harvesting complex. Meaning that for each complex, there are many kind of pigments. There's a chlorophyll A, chlorophyll B, pigments such as carotenoids, xanthophyll, and so on. Okay, so if the excited chlorophyll following the third destiny, this energy is passing to the next molecule surrounding, and they can play ping pong with it. They can play ping pong, and every time they play ping pong, the energy get released. Okay, kind of like the tuning fork. You know tuning fork? You know tuning fork? Oh, what's in Malay? I know we found out the word in Malay. Tala bunyi. You have the tuning fork. You bang it against the table. It's you touch upon. It's going to cause rippling effect, right? And then you can touch the second point, the third point, the fourth point, the fifth point. But as you are touching more points, what happens to the ripple? Is it going to get stronger or weaker? Yeah, so that's the concept of number 2 here. As the chlorophyll transfer the energy to the next molecule, the rippling strength gets lower, weaker, and until it becomes ground state again. As simple as that. Number 4, what happens with excited chlorophyll? It can become a problem. The chlorophyll become an alien. It's not passing around the energy. It gets excited. This is your friend laughing until Deepavali. Yeah. It becomes this thing. Chlorophyll star 3. This thing is called, what's the name? Yeah, triplet excited chlorophyll. Triplet. So that's why it's got a 3 in it. Okay. So why this happens? You're talking about nature now. So don't expect anything, because everything is possibility, probability, and possibility. Okay, you learn about electron valence, right? In the atom or molecule, the electron they are talking about in the orbital 1, 2, 3 is just a probability of it being there. It can be here, it can be there, but it's still within the region. It won't go beyond that. So it's a probability of it being in one place or another. So if it following this state, this way, it will become the triplet chlorophyll. Okay, damaging. Okay, triplet chlorophyll will cause the formation of ROS, reactive oxygen species. This thing in our body is the thing that is causing cancer. Yeah. In plant, well, the plant's not going to get breast cancer, but you're going to break havoc on the membrane. Okay. There are many types of ROS, reactive oxygen species. This radical oxygen is one example of it. And number 5, excited chlorophyll can actually do photosynthesis. Okay, so get excited, and this energy is passed to the first electron acceptor in the electron transport chain. And usually the first acceptor is called phyophytin. It's not written there because this should be a lesson, but I'll just tell you anyway. phytin. So can you see that? Number 1 until number 4, it has nothing to do with photosynthesis. To tell you that if you want, I just want to relate with your agriculture mindset. Imagine if you can give a treatment to your crop or do something to your crop to reduce the probability of 1 until 4 from happening. You increase the chances of your crop to do number 5 exclusively. That will tremendously boost the photosynthesis, and by positive correlation, you will get higher yield as well. Just simply by changing here. And there are many things that can happen here. For example, by giving nitrogen. Extra nitrogen helps with the formation of healthy pigments. Okay? And that can actually support the plant to do more photochemistry rather than releasing all the energy back randomly. Okay, right. What time now? 10 minutes, 10 minutes. Okay, so about pigments. So the pigments in plants, it's actually, there are many kind of chlorophylls. Okay, I just need you to understand that chlorophylls, I think up to now people found out until E, A, B, C, D, E. So in plant, we are mainly deal with chlorophyll A and B. C, D, and so on, chlorophyll in other organisms. Okay, so the point here is there is a chlorophyll in plant. There is also bacterial chlorophyll. Chlorophyll is not only present exclusively in the plant, so please correct that understanding. Okay, there are many other of organism in other kingdoms that actually photosynthesizing. Okay, they got chlorophyll in the body, but not to necessarily, producing fruit or anything. It's just part of the mechanism, the ancient mechanism of it. Okay, and in addition to chlorophyll, you can also find other pigments such as carotenoids and billin. Carotenoid is you can see in not orange, carrots, and billin. Billin is actually, I think, red. Phycoerythrobillin. You usually find this in red alga. I don't think it's written there. Yeah, you usually find it is in red alga. Yeah, and these are, remember the spectrophotometer, the one with prism. You can control the wavelength. You can use that to ID the absorption spectra of any of these pigments. You can see that there are many pigments: chlorophyll A, chlorophyll B, bacterial chlorophyll A, bacterial chlorophyll B. Each of these has a signature absorption spectrum. So this is how scientists know the extract is what compound. Is it chlorophyll? Is it carotene? Or is it something else? This is how, because it's got a signature, and this is highly conserved throughout the kingdom. Even if you go to other countries, it is the same. Okay, so you can see that most of it got what we call as a double peak. For example, the bacterial chlorophyll here, it got the peak around UV. It's absorbing UV. Okay, anything before 400 nanometer, it's UV. UV got 3: UVA, B, C. The lowest is UVC. The highest is UVA in terms of energy. Right. It got the peak here for bacterial chlorophyll A, and then it got the peak again somewhere here. Well, let me follow this. I need to. Yep. So this is beyond 700. This is far something that you cannot see. Okay. No wonder bacteria is very successful. You are avoiding sunlight, UV, right? You're wearing sunscreen and everything, hoping to be beautiful forever. But bacteria is utilizing the UV light to thrive. Yeah. This region is actually kind of heat. You are shying away from the heat. Bacteria is using the heat to, well, to utilize, to get the chlorophyll excited. Yeah. So if you think about it, next time when you are cooking up your Maggi in the kettle, you feel the heat, right? Or maybe your friend got fever. Remember, heat is light. Why you can't see it? Because it's in this region beyond 700. Your eyes cannot see it. The proof heat is light. Actually, you can see this. You boil your water, the kettle on the stove. Next to the boiling pot or kettle, you start to see the image kind of look blurry somehow. That actually your brain is trying to make sense what's happening. The wavelength, maybe 800, has hit your retina. But your retina do not have specialized cells to interpret this signal. So your brain kind of confused. I cannot interpret this. So all the best it can do is make the image look wavy. Yeah. Kind of like you are feeling like something in there. Actually, there's a hantu behind anyway. Yeah, you can feel the presence, but you can't really see it, but you know it's there. Yeah, kind of like that. Okay, and so I remember talking about the carotenoids, xanthophyll, and so on. These are collectively known as accessory pigments. Okay, there are many other pigments here. Okay, in the plants. That's why you look at the plants. Some plants, even if talking about the green, there are many shades of green, right? There are darker green plants, lighter green plant, yellowish kind of plant, hijau pisang, hijau pretonas, hijau kuih talam. So many shades of green. Why? Because chlorophyll is not the only thing present in the leaf. The interaction of chlorophyll with other pigment molecules will determine the eventual appearance of greenness to our eye. Whether it's going to look bluish, yellowish, or maybe it's going to look like transparent. Yep. Some leaf, no, well, not really transparent, translucent. Some leaves do look translucent. Okay, that's something for you. To read. Okay. I think this is finally for this. Yeah. I just put it here so that you can have some appreciation what kind of chlorophyll is present in other organism. Okay, eukaryote, under eukaryote, you got your mosses. Mosses is lumot, green algae, euglenoids, diatom, and so on. And then you got prokaryote, like cyanobacteria and sulfur propylbacteria, and so on. Okay, yeah. And a little story about the hemoglobin versus the chlorophyll. Therefore, sila jangan anti chlorophyll, makan sayur. Okay? Yeah. Do you know how much we need to eat actually, the green leafy vegetable, in order to be healthy? Do you know how much? I think they did a study about this actually. Mainly actually to get the potassium out of the green leafy vegetable. It's about your hand. It's like a cup, right? You need about 6 to 8 cup of your hand per day in order to stay healthy. Yeah, that's how much. Yeah, it can be any salad. It can be choy sum. It can be lettuce. It can be anything. But that's how much actually. Yeah, because remember, you are not feeding you. You got many microbiome babies in your tummy. I want to do that again. Tapi tak reti. Macam mana? Ha, macam tu. Yeah. Actually, all this modern diet is hurting the population greatly. Okay, even the vegetarian is not a healthy choice anymore because the agriculture produce that they are eating is not nutritious compared to 10-20 years ago. Okay, all the plants, crops that are being grown now, highly the product of commercialization. It has been geared towards high produce, don't care about the nutrient. As long as you get the sweet, you got the heavy, that's what people want. Okay, so what can you do? I don't know, makan lah. Rule of thumb: if you can achieve 4 to 5 colors in a day, you are good. You good anyway. For example, today you decide, okay, I'll have green. My green is my sayur bayam or anything, and then I have my pumpkin. Pumpkin, there's another color as well. Then I have a fruit. Maybe you want I don't know, tomato. Ada korang makan buah as tomato. Yeah, dragon fruit. Yes, that's another color. And then you got 2 color left. Don't go to cheese ring. That's that's not healthy color. What color? Yeah. Purple, mango. Yeah. Or nuts. Nuts actually got color as well in it. Yeah. Not all color looks interesting to your eyes. Okay, some colors they have the color, but they don't necessarily look interesting to our eye. Okay, I think up to this point, we'll continue this. I just want to highlight the schedule for this lesson. If you can turn on, open this. Can you open this one on here so that people can see? Is it in here? The timetable. Okay. I actually want to start this early. Okay. So my class. So let's start early so that you can finish early. All right. So the plan is to start at 7:45, 7:45, and then we'll finish by 9. Okay. The reason is I need to prep for your experiment because the experiment is meant to be on the same day. So I need a bit of time to do that. Okay. So for make it smaller to kau ni. Okay. So in essence, this is the lesson plan for tengok aku jadi chlorophyll excited sekarang. Tak apalah. I don't want to do any replacement class. However, you got so many Mondays holiday. That's not healthy. Okay? Because last year I got 6 or 7 Monday with holiday that kena holiday. You are happy about it, but actually that's not healthy because this is a time for you to learn. When else you have the time to learn, right? So take this opportunity because I can assure you when it comes to photosynthesis, up to this point, no other university is offering this tip. Memang sebab aku yang ajar. Right. Okay. So for today, we'll do. So we have filled out. We need to make plan kejap lagi. So you will only have one experiment. You see, I have designed it to make it so easy. There's only one experiment. So we are going to use rice because we have rice experiment going on anyway. So in essence, your lab petang nanti is ladang oriented. So please don't look pretty. Yeah. Wearing umbrella so on, I don't mind. You know, some lecturer kind of crazy. Tak bagi pakai payung. No. You can bring umbrella. You can bring fog leaf upon you. Okay. Go ahead. Okay. So we'll try to follow as what is written here. If you see 9.1, 9.2, 10.6 and everything, that's actually corresponding to the lesson plan here. Okay. And guess what? I need to respect the time. So if for some reason we are not able to finish the lessons, it's your responsibility to read the lecture note. Lecture note will be given, don't worry. Since I can see that our students do not like to open the book, don't worry. I have turned your book pages into slides now. It's very easy. So even if you don't open the book, and hopefully you get the most, you just need to read. Okay? Because I've been reminded by the meeting, the faculty meeting, to increase SCL, especially for these 4,000, 5,000 courses, meaning that I only teach some of it. The rest is for you to read. And after you have read, I have put a little exercise at the end. Okay? You see exercise? There's an exercise there. So that's actually for you to self-check. And when we see again the next coming lesson, we'll see what's the answer like. So you can do the self-assessment. Please do not throw this away. Keep this, because this will be your carry mark. You see, it's actually possible to get full mark, right? Yeah. The trick is, if you answer anything, make sure if suddenly I call you, you can show from the notes where is the answer is located, which paragraph, which page, right? Okay, do it yourself. Okay, it's okay to discuss, but you need to understand yourself. So do this from the beginning. It's still early, first week, first semester. So have the right mindset, have the correct attitude, you'll be fine. You'll be fine. I know it's a lot to read. And the reason I give all this lengthy thing for you to read, because most students are actually kind of like late bloomers. You need time to ripen. You look one time, you still cannot absorb it for quite some time. Maybe you're not sure about some of the words. I need to open dictionary for this. And then you panic. And then whatever you have learned, all forgotten. Right? So take your time to read. Within 24 hours, revise again. Okay? There is no other way. Like, really, small discipline now can set you off for life. Like, really, have it embedded. Okay? So do not tell the lecturer it's not helping. I can assure you, not the subject bagi kau nota macam ni. I just want to assist you. Okay? So coming back to here. So for the next week, you can see that before the farm, we will have the lecture. So most probably, we'll have the lecture in the physiology lab. Brief lecture, okay, to keep in time with our lesson schedule, okay. And then we'll go to the farm, which is about under 10 minutes walk from here. It's not very far. Then we'll do the progressive checking on the plants, because now you're growing plants. You need to do all the agronomic practice. You need to do the fertilizer. You need to do the pest control, right? Can I close the SMP so that nobody can come in? Do I have that power? So you can have your group to do that. Okay, right. We'll have the holiday. Pay attention to the holiday. Bear in mind, even though it's holiday, now you have your plants. Have assigned somebody to look after the plants. Okay, it's not like you have to spend 1 hour, 3 hours straight. Okay, have a regular checkup on the plants, and I'll tell more about it in the afternoon. Okay, right. Okay, just that. Any question? Yep. Anybody want to drop? You can say drop. Yeah, within 2 weeks of the semester opening, if you drop, you don't get fined for it. All right? Okay? Right? Yeah. Bagus kan kelas sikit semua orang boleh dapat nota color. Okay, any question? If not, I think that's all for now. So I'll see you in. Oh, by the way, I think before we'll have Aku kan macam tak nak cakap dengan orang kan. So we'll have the, wait a minute. I want to use the Google Space. Tak pernah guna benda tu. But I need to invite you first. I have invited you. Kau dah masuk belum? Kenapa tak masuk? Stupid lah internet ni. No, I need to invite you. You need to put the rest of you need to put